Hello, everyone, and welcome back to another episode of Neurosci IQ. In this episode, we will be talking about Parkinson's disease. So, let's get started. First, let's talk about the symptoms of Parkinson's. The most well-known symptoms are the motor skill symptoms, for example, hand tremors or difficulty walking or postural instability or rigidity. But something that you might have not known is that Parkinson's also causes non-motor skill symptoms, especially in the later or the advanced stages of the disease. For example, people with advanced stage Parkinson's could display signs of cognitive impairment such as memory loss, depression, a loss of sense of smell, or also joint pain. So now let's do an overview of Parkinson's disease. It is basically characterized by the loss of dopamine neurons in the substantia nigra. In fact, in some cases, up to 70% of the neurons in the substantia nigra have died. It is important to mention that of course other neurons like other dopamine neurons in other areas of the brain are also impacted, but for the purposes of making this video simple, we will just focus on the neurons mostly in the substantia nigra. And as you might have already known, there is no cure, and most of the treatments focus on the management of symptoms such as hand tremors and pain. And while the causes are unknown, scientists believe that both environment and genetic factors likely play a very important role in someone getting Parkinson's. So if you look at the diagram, you can see, for example, here is a diagram of the substantia nigra in your brain. And if you look at the region, we have the pigmentation, and that's how you know there are like a lot of healthy neurons when you see the pigmentation. However, on the right side, we have the substantia nigra of a Parkinson's patient, and you can see there is not a lot of black dots or pigmentation, which indicates neuronal loss. And if you look at the zoom in version, for the healthy patient, we have a lot of neurons, and they are secreting a lot of dopamine into the synapse, which a healthy individual should do. However, in the case of a Parkinson patient, you can see that one, there are fewer neurons in the first place, but these neurons have been impaired, and we will get to the reasons why later, and they aren't secreting as many vesicles. So as you can tell, Parkinson's disease is primarily characterized or caused by a loss of dopamine neurons in the brain. So now you might be wondering, well, what is the role or importance of dopamine in the brain in the first place? Dopamine plays a wide variety of functions, and there is a lot of different pathways or areas of the brain that actually use dopamine. While, as I discussed, Parkinson's disease affects multiple areas of the brain and multiple regions of the dopamine neurons. But the most important part to remember for the purposes of this video is the nigrostriatal pathway, which is responsible for voluntarily movement production. So while it impacts other areas such as the mesocortical pathway or the mesolimbic pathway, we will just focus on the nigrostriatal pathway because that one seems to be the most impacted one, and there is most of the research that has been conducted focuses on the nigrostriatal pathway. And as you can see, for example, dopamine neurons play, of course, a role in voluntarily movement, but also cognition, learning, motivation, emotion, and even addiction. So as like a patient with Parkinson's, all of these areas will be affected. So now we can talk about the important or the hallmark features of Parkinson's disease. And there is basically three main parts that are super important for us to know. Basically, we have toxins and protein aggregates that build up for reasons that remain unclear in the neurons, and as these proteins build up and build up and build up, it leads to neuronal loss. Specifically, the dopamine neurons are the most affected and they start degenerating. And as you might have known, most neurons in the brain do not come back, and if you lose them, then there isn't a lot of replacement that can happen, and you could imagine why that's very bad for you. And after these neurons die, there is actually the immune system, and the microglial cells can come in to recycle the material and all of those things, but they cause a very strong inflammation response. As a result, there is a lot of neuroinflammation, and this causes the blood-brain barrier to also be impaired, and it makes the blood-brain barrier leaky. So now that the blood-brain barrier is leaky because of the inflammation, it causes the other toxins and other harmful chemicals to reach the brain, which they also cause more damage to the brain. So as you can see, neurons die, the blood-brain barrier becomes impaired, then more toxins, more harmful chemical can enter the brain, and the cycle keeps on repeating and repeating and repeating until there is almost like all of the neurons have been destroyed, unfortunately. So now that we have discussed the exact roles of the dopamine neurons, you might be wondering, well, how do they exactly die? We mentioned the toxins, for example, and the protein aggregates, but now we are going to discuss them a little bit more in depth. So while there are a lot of reasons why dopamine neurons could die, in this video we will just focus on the three most well-known ones. The first one is dysfunctional mitochondria in the neurons. The second one is protein aggregation, specifically alpha-synuclein, and also there could be a genetic defect in the protein recycling mechanisms in the neurons, which could also lead to protein aggregates building up and causing the neurons to die. So now we are going to talk about this concept a little bit more in depth. First, let's talk about the mitochondrial significance in Parkinson's disease. As you already probably know, mitochondria is known as the powerhouse of the cell because its job or main function is to generate ATP or energy for the neurons to function. And in some cases, there is a lot of that for some reason, some mitochondria are highly susceptible to environmental toxins or other environmental effects such as radiation, and these toxins or other environmental factors can damage the mitochondria. And when the mitochondria becomes damaged, then as you can tell, it basically starts falling apart, as you can see from the diagram at the top. And when the mitochondria starts dying, this leads to a wide variety of events and eventually leads to the neurons dying because one, they do not have enough energy or ATP to support the cell, but also there is a lot of chemicals that are released that, for example, increase the oxidative stress inside the neurons, and the oxidative stress causes a lot of proteins to build up, especially alpha-synucleins tend to build up when there is a lot of oxidative chemicals in the cytoplasm of the neurons. There is also genetic factors that cause the neurons to die, especially in Parkinson's disease. The two most well-known genes are the PARK7 and the PINK1. So the PARK7 basically encodes for a protein that is tasked with breaking down reactive oxygen species, also known as ROS. And what Park7 the gene does is encodes for a protein. And if, as you can tell, this protein is breaking down the ROS into other parts. And what happens is this causes the ROS, or the reactive oxygenated species, to not be highly reactive anymore and not cause a lot of damage to the cells. So a normal Park7 gene would encode for a protein that is very efficient at breaking down oxidative agents in the cell, which reduces damage in the neurons. However, in some individuals who are more prone to getting Parkinson's disease, unfortunately, this means that ROS cannot be broken down, and it keeps on building up in the cell, which damages the neurons, and the dopamine neurons, for example, die, leading to Parkinson's disease. Another significant gene is the PINK1 gene, and this gene is responsible for making sure that the mitochondria are functioning properly, and it encodes for a protein that allows the cell to detect these abnormal mitochondria, and the abnormal mitochondria are targeted for degradation and recycling. For example, if there is an abnormal mitochondria in the cell, then that mitochondria might produce a lot of toxic substances such as reactive oxygenated species, which are highly toxic to the cells. So if these mitochondria aren't recycled, then it causes oxidative species to build up in the cell, which leads to neurons dying. So people, for example, with defects in Park7 and PINK1 genes are at a higher risk of developing Parkinson's, because unfortunately they cannot get rid of these very oxidative or reactive species, which leads to the neurons being more likely to die in the first place. Now let's talk about actually the formation of proteins or formation of aggregates. Essentially, as I discussed earlier, Parkinson's is characterized as an alpha-synucleinopathy, and what that means is that this protein in the neurons that is very important for their function, as a monomer, it's just floating in the cytoplasm, but because of some conditions such as toxins or oxidative agents, it causes the protein to misfold and form a lot of aggregates. So it keeps on getting associated with other misfolded proteins, and it keeps on getting bigger and bigger and bigger and bigger, and it forms insoluble large protein complexes known as Lewy bodies. And the problem with Lewy bodies is that it impairs neuronal function, because these are kind of toxic to the cells and take up a lot of space. Also, another thing to remember is that alpha-synucleins have also been implicated in other diseases such as dementia and multiple system atrophy, among others. So these protein aggregates cause a lot of problems in the cell, and these are what is the main reason for Parkinson's disease, the buildup of alpha-synuclein monomers. So now you might be wondering, so for example, if these proteins form aggregates, why does that matter? For example, maybe one neuron that was not functioning properly might die. But unfortunately for us, these proteins that form aggregates can actually spread quite easily to other neurons. For example, it has been hypothesized that these aggregates can cross the synapse and spread to other nearby neurons. So essentially, if one of the neurons dies or is implicated by these aggregates, then these proteins or alpha-synuclein aggregates can cross and enter other neurons, whereby they cause other proteins to misfold in the nearby neurons, and the nearby neurons also end up dying. So it kind of is like an infectious disease. It's just that one neuron can spread it to other neurons that are nearby. And for example, a single neuron could be in contact with up to 10,000 additional neurons. And that causes, for example, one neuron to be able to transmit these aggregates to so many other neurons. And it keeps on progressing and progressing and progressing until there is very few dopamine neurons remaining in the brain. So that was it for this video. Thank you so much for watching. If you enjoyed, please make sure to like and subscribe our video for more content.